Hello everyone, and greetings from a dark and rainy Gothenburg in Sweden. If it's, I'm very happy for everyone that can participate now. I'm Carl-Frederik Burman, or Kaffe, and my involvement in the Ideal project has been through the Chalmers University, but I'm normally working at AstraZeneca, so I'm a bit of an industry contact within the project, bringing in that kind of perspective. So today I will talk about the decision analysis work package, and we will have a view where we are looking at the different stakeholders that are important to bring innovative drugs to the patients in need. So important stakeholders today will be the companies, the sponsors that are developing drugs. It's the regulators approving, checking if drugs are efficacious and safe to be permitted on the market. And it's the payers, the reimbursers. And of course, in the background, we have very, very important stakeholders, the patients and their prescribing physicians. But we will not talk very much about them today. So the Ideal project, if you have followed this series, you have heard pretty much about this before. The Ideal project has a lot of different work packages and spanning a pretty broad area. So my work package has been decision analysis, and we have been having a number of collaborations, and one of the collaborations that I will touch upon a bit today will be on the fourth work package about adaptive designs, which is led by Franz König at the Medical University of Vienna. The advisory board have been covering a lot of different perspectives, and somewhat to my surprise, it's two economists that have been doing a lot of good work in collaboration with us. It's Martin Forster and Paolo Pertile. Then also we have a lot of good statisticians and representatives from patients, the rare disease community, and so on. So much of the work has been done by Sebastian Ljubjörnsson, who is a PhD student at the Chalmers University. We have, as part of the work package 9, we have also had Frank Miller at Stockholm University. And much of the key example, I will say, of this presentation is from the collaboration with Frank. We've also had Søren Christensen at Chalmers, and then we have Paolo and Martin from the External Advisory Board, who has been contributing to one publication. We had the collaboration with Franz König, and also with a neighboring project, the Inspire project, doing similar things as we. So we have one publication together with Franz and Thomas Honda, Martin Posch, Nigel Stallard, and a leader from the DIA, Adaptive Design Scientific Working Group, Bob Beckman. And he's also standing a bit for the medical input to the work, as he is trained as an oncologist. What we have been interested in this work package about decision analysis, it's mainly about how different stakeholders make decisions on the key things that are important in drug development or late stage drug development, but after drug development also going into the market, so it's the pricing decisions involved here as well. And as I said, the key stakeholders for our models would be the commercial sponsors, the regulators like EMA or FDA, and the payers. And the payers look quite different in different countries, in different regions. But in EU, it's often a single payer in each country or part of a country. And we have been using NICE, looked specifically into NICE in the UK. And we're interested in how are these stakeholders make decisions, but more than that, prescriptive, how should they make decisions to get the best output to the society, to the patients. So we are thinking about how to set up incentive structures or how to align different stakeholders. And the applications are on orphan drugs and also on subpopulations, biomarker-defined subpopulations. The methodology have been drawing from different areas. So I'm a pharmaceutical statistician. I've been working in the industry for more than 20 years. So it's pretty much of standard pharmaceutical statistics thinking. But we're adding Bayesian decision theory, as what we are doing is very much about making decisions. And I think, interestingly, in the statistical world, we're trying to bring in an economics perspective. And that's partly about thinking about agents as rational, profit maximizing in many cases, or maximizing the utility for all patients. And from economics, we have also taken the concept of mechanism design, which is something that I will show in an example. So the generic model is, I've chosen not to go so much into the specific examples, each and every publication or a case study. I choose to present a general model that's rather abstract. And it's abstract not to be more theoretic or more difficult, but rather to make it easier to understand what we are working on. And you can always check the details if you're going to the publications. So what I will present is pretty much the generic model is spanning all what we have been doing in different sub-projects. So let's start with the sponsor's decision regarding phase 3. And this decision is, first of all, it's a go-no-go decision, an investment decision. Should a company spend a lot of money to check if this drug is to prove that it's efficacious and safe and potentially get a market authorization? It's if the company makes a positive phase 3 investment decision, and it's a matter about the kind of statistical design of the trial. So that includes picking the sample size and perhaps not so naively as looking at the textbooks, just plugging in theta value and an alpha and beta and sigma, and you're finished. Much more of looking at this as a real investment decisions, where we have a cost for in terms of money and time if you're increasing the sample size. But of course, we have a gain in terms of higher power. It could be also questions about which population should we run the trial in. And in one of the publications, we are looking into the problem about having a biomarker positive, biomarker negative subpopulation. Should both these populations be tested, be included in the trial, and how should we analyze the data to potentially get market authorization in the full population or only in one of the subpopulations, the biomarker positive one. This could also include questions about multiplicity, corrections, doses, etc. I will not talk about doses today, and you could imagine a number of other design features that we will not touch on. So DS is the sponsor's decision, and this is including everything I was talking about. So you can think about mainly the sample size, and putting the sample size to 0 will mean that we are not running a trial at all. So that design will, that trial with the design DS will give data X, and of course data are random outcomes. Included in the data we have the efficacy, the estimated efficacy of the drug, but it is also safety variables, so it could be very multidimensional. We will concentrate now on the efficacy part, but it's kind of straightforward with the same kind of concepts to look at, for example, clinical utility for the patients, where we are putting efficacy and safety together. And I will mention something about the uncertainty in how regulators view efficacy and safety later on in the presentation. So the data X are reviewed by regulators, and based on the data and the design generating the data, regulators will choose whether or not to grant market access to this drug. Perhaps I should say that in the design DS of the this is the design of the phase 3 program, and for rare diseases that may be one single phase 3 trial, but it could, and it's standard for common diseases that this is 2 trials. Often these trials are more or less replicates of each other. We are typically thinking about the market authorization decision as a 0-1 decision, dichotomous decision, but it could be, for example, restricting the approval to the biomarker positive subpopulation. Payers will, given a regulatory approval, will also look at the data and decide if they would like to pay for this, reimburse the drug, and in that case, which price that they would accept. And we will especially look at NICE, so maybe the payer or health technology assessor, which is clearest in terms of laying out how they are looking at this decision and how much they would like to pay. And they are thinking about in terms of quality adjusted life years, and how many pounds to pay for every QALY. QALYs could be questioned in a number of different ways, but this is what we will be using in one of the what we have been using in one of the publications. Then, if we have all this, we have this structure of the sponsor's decision leading to data, and then regulators, payers making decisions on this. All this will eventually lead to a potential reward for the sponsor. So, or here we're thinking in economic terms. And the net reward will also depend on the cost for the clinical trial and the time. So the expected net present value for a company may often decrease very, very rapidly. So 1 year's delay could mean that we are losing a decent share of the potential revenues, especially if you have a strong competition on the market. So for rare diseases, this may be more of getting first to the market, getting market exclusivity, but the time is always important. Putting a bit more of statistics into this, of course we have a parameter behind the data that we are generating. So we don't know theta here, and theta is governing everything you can think of. So it's multidimensional. And it could be more factors in this. So it could be that different parts of the population have different efficacies, and all this is part of our parameter or parameter vector theta. So of course we don't know this from the beginning, and one possibility is to model this in a Bayesian way, so put a prior for the parameter for prior on efficacy and safety and whatever you would want to incorporate in the model. If you don't like to be a Bayesian, you can always put the prior to be a 1 point prior. So you say that assuming that theta is theta a fixed value, what is happening then? So let's look at this complete model now, this rather abstract model. And now we see that the sponsor decision, DS, is eventually leading to a reward R. And we have been putting in a parameter on the prior, so we know for every DS we know the distribution of X, and if we understand the regulatory rule and the pay rule, we get a distribution for R. So we can calculate the expected value of the net reward R. So E of R, the expectation of R, is a function of the sponsor decision. So say now that the sponsor is rational in the sense that it aims to maximize the expected net value. Then the sponsor will choose the decision, which we are marking with a star, DS star, to get the highest expectation of R. So DS star is the DS, the decision maximizing. So if you are only looking at this, then there are a number of publications about the optimal sample size. This is taken from one of the publications from the IDEAL project, and I will not go into the details here, and there are a lot of papers you can check. This is kind of the same thing, but now we're looking at 3 different types of design for a problem where we have 2 biomarker-defined subpopulations. So we are comparing a stratified design where we are including both biomarker positive and biomarker negative patients, and we may win in the biomarker positive group or in the full population. An enrichment design where we only go in the biomarker positive group that we think may have better effect, and a classical design where we are not even testing the biomarker status but taking everyone into the trial and only analyzing the full population. And here we can see the optimal sample sizes in one example where on the x-axis we have the prevalence of this subpopulation. And to this we can also add the utility functions and compare these 3 different designs, which one is best and which sample size is best for that kind of design. So moving back to our generic model, I would like to add something about the uncertainty in regulatory decisions, payer decisions. So in the kind of implicitly, we may have been thinking about the R and the P here as deterministic functions. So if you know the data, then you would know the regulatory decision and the payer decision. And of course, this is not the case. There's empiric data that we have been using showing that NICE in the UK, and I will say that NICE is comparable compared to other payers, quite clear about their decisions and what's underlying that. But there's empiric evidence that there is a pretty large uncertainty still about which price that NICE will accept. And it's also quite clear that investors say that has been seeing at least the high-level data from trials cannot really predict always what the regulatory decision will be. And in advisory committees, you may have a split vote when some people think that FDA should approve this drug and some think they should not, or some think that it should get a restricted label. So it's not so obvious, and these functions are not really deterministic and well understood to the sponsor. So what the sponsor can do is to try to model how will regulators, payers react on a certain data set, and doing this before generating the data. So that means that trying to figure out the distribution for possible the probabilities for different decisions based on all possible data sets. But in principle, we can average out over all this, take the expectations over the regulatory payer decision rules that we are now seeing as uncertain, stochastic if you want to. And then what we can do is to compare what will a rational sponsor do if these rules are not certain compared to what would a sponsor do if we had deterministic rules. And then we see that there is a clear cost for the sponsors and at the end for the patients. So the patients will miss some useful drugs. They will never come to the market. They may not even be tested in phase 3 in some cases, because there is an uncertainty about how will regulators look at benefit risk, how important is safety compared to efficacy. And for payers, how will they assess data, efficacy, safety, and what price would they be willing to pay. So examples here is from Sebastian Jobersson's licentiate thesis, where he has one example about how the value for the sponsor is looking when you have certainty or uncertainty. And he's quantifying how much you gain if you have full knowledge about the regulatory decision rule. And this is about weighting efficacy and safety together. And a full publication focusing pretty much on the uncertainty in NICE decisions. And here we are using economic concepts where we are looking at the demand, which now is the expected demand, the expected or the probability that NICE will accept a certain price. And we can put this into a standard economics framework where the probability here is playing the same role as demand is playing in standard economics. And we can look from that probability to expected profit and the optimal price for the sponsor to suggest. So now to mechanism design. We have been talking about how rational sponsors are behaving, but how should a rational society act? And society now consists of the regulatory authority and the payer. So take a very, very simple example here. This is the distribution of the efficacy of a new drug. And the scale here is chosen so you could think about this as hazard ratios for oncology drugs. And then we have been thinking about what is likely, how likely are different effect sizes in cancer. So I think that if you're looking at drugs moving into the first efficacy testing in phase 2, then I think that that some of the drugs being tested do not have any effect at all. So they will have a hazard ratio of 1. And in the publication together with Frank Miller, we have assumed that a certain percentage of the drugs have no efficacy at all, hazard ratio of 1. I'm taking away this point mass in hazard ratio equal to 1 for simplicity. So I'm only drawing the prior distribution for all other effect sizes. So here, everything below 1 is having a positive effect. It's increasing the median survival time. However, there's typical toxicity or some safety issues with all kinds of drugs. In oncology, they are often doses of a pick to get a certain amount of toxicity in phase 1. So think now that a hazard ratio of 0.75, a drug with hazard ratio 0.75 for progression-free survival, that's borderline useful. So the efficacy gain here and prolonging the median survival by perhaps 1/3. It's the same. It matters as much as the toxicity cost for this drug. So when the hazard ratio is 0.75, the gains in terms of efficacy and the cost in terms of toxicity, they are equally important. So the clinical net utility for the patient is 0. Drugs with hazard ratios above 0.75, so 0.8, 0.9, and of course 1, they will have too much toxicity. They should be rejected, while drugs with hazard ratios below 0.75, they are useful. So now, as a simple example, we are putting all these drugs, a lot of oncology drugs for indications with similar properties, so the same population size and the same cost of clinical trials, the same sample size in phase 2 we will pick for simplicity, and the same benefits for the sponsor if the drug is succeeding. So take a lot of drugs, say 1,000 drugs or whatever, which is forming the black curve here, and we put everything to the same kind of phase 2 trial, and we have a standard criterion for the sponsor to continue or not into phase 3. And then we get the red curve here. So the sponsor he's here rejecting the most of the drugs that has no efficacy, so it has a ratio of 1. But still, due to the randomness, the stochasticity of the data from phase 2, the drug that has a true hazard ratio of 1 could get an estimate of 0.7 and look as a good drug. So we have here a certain error in terms of a small proportion of the drugs, perhaps 20%, going further to phase 3 testing, even if the effect was 0. And we see here that at 0.75 we see that a little more than half of the drugs are taken further to phase 3 testing. And if the effect is very, very large, 0.45 as the hazard ratio or even lower, then we are more or less taking all the drugs. They get to phase 3 go decision and we move on to phase 3. So the red curve is the distribution of the drugs that are moving on to phase 3. So we are starting with the black curve, which is exponentially distributed in not the hazard ratio scale, but the minus log hazard ratio scale. So we are starting with this prior. We are putting all drugs to a phase 2 test, and then we have a certain cutoff, and the drugs that are moving on to phase 3, they will follow the red distribution. And this is not a probability distribution. This has a total mass that is less than 1, so it's a sub probability distribution. Okay, so back to the generic model. We have been talking about the optimal decision for the sponsor as maximizing the expected reward, net reward. So that means that we can see that we have an arrow back from the reward to the optimal decision. And when looking at this model, we see that the reward is also dependent on the regulatory role and the payer role. So we can say that that actually the optimal sponsor decision is a function of the society's decision roles. So the market authorization, regulatory role, and the payer role. And that means that for certain roles from the regulator and payer, we get an optimal decision, a red curve for the distribution of drugs going into phase 3, and for this regulatory role, the green curve will be the distribution of the drugs that the sponsor is taking into phase 3, and then regulators are accepting based on the stochastic data in phase 3. Now we could, since the optimal sponsor role is dependent on the regulatory role, we can think about what is happening if you're changing the regulatory role. So if you are looking at a reward for society, and you could think about how much is the expected health benefits for all the patients that could potentially get this drug. And this is net utility, so it's efficacy minus toxicity properly weighted. You could also factor in here, you should probably have to factor in the cost for society. But we get a net reward for society, say an economic value in terms of health benefits minus the cost. So what we can try to do is to optimize the regulatory role or the payer role to make the optimal outcome for society. So if we go back to the curves that we had before, the black line showing the distribution of drugs before phase 2, the red curve is the sponsor's decision based on the old regulatory role. This is the distribution of the drugs going into phase 3, and the green curve is the distribution of the drugs also having good enough efficacy according to regulators. So having an estimated in phase 3 better than a certain value, or being statistically significant at a 2.5 significance level or something like that. So the green curve is the distribution of drugs accepted, and this is based on a certain regulatory rule leading to a certain sponsor rule. So now let's change the regulatory rule. Let's make the regulatory rule somewhat more liberal. So this could, for example, be that we are changing for a rare disease, for a rare cancer, we are changing the alpha level for regulatory testing from 2.5% to 5%. Say that will mean that more drugs will be accepted, and we are changing the curve now from the solid green to the dashed green curve. And in this case, you see that we get more drugs out of the patients. However, some of these drugs are making more harm than benefit. So the difference between the two green curves in the area where the hazard ratio is higher than 0.75, then the toxicity will be higher than the efficacy, and these drugs giving a negative utility to the patients. Whereas on the other side, hazard ratios below 0.75, any increase from the solid green to the dashed green is a benefit to the patients. But the regulatory rule, changing the regulatory rule will change the incentive structure and change the optimal decision from the sponsor. So the red curve now is changing to a dotted red curve. And it's that dotted red curve that will be the distribution of drugs going into phase 3 and then tested with a new regulatory rule, which means that the net will not be the dashed green line, but the dotted green line. So we see that we have to factor in how sponsor will react on the regulatory decision. Changing the regulatory decision in this way will mean that we are going from the solid green to the dotted green line. And the difference there is this purple line, and we can think about, is this giving more benefit than harm? Is this getting more useful good drugs than bad drugs? And in this case, it's pretty much zero extra utility. So these decision rules will get the same public health benefits, more or less. And we can tailor this to get a rule that is optimizing the total patient utility or the total societal gain in factoring the cost of the drugs. So this is some of the details given in the Miller paper. And these are examples from that paper showing something similar, but also getting into the situation where we have drugs with hazard ratio exactly 1, or in that paper a true efficacy of 0. So something about pricing, which is the topic of one of the other publications, together with Sebastian Björnsson and the two economists, Martin Forster and Paolo Pertile. And this is based on empiric data for the UK payers, and it's not the whole of UK. I think it's England and Wales, which is directly following NICE recommendations. But NICE recommendations is something that people are looking at other reimbursement agencies as well. And this is showing here that the optimal is shifting when we're changing the population size. And we can also say that we have an increased, that the uncertainty involved in the payroll is something that has a quantifiable cost to sponsors and to the society. So summing this up, we see that rational sponsors are basing their investment decisions on the things that will be important to the potential success and the potential reward for the sponsor. And that's very much the incentive structures that the society is building. For orphan drugs, there are specific incentivizing mechanisms, but it's also the regulatory decisions about whether to accept or not accept a drug. And the regulatory decisions, we can see empirically that regulators are more liberal for rare diseases and very rare diseases than for common diseases. However, I'm not really aware about FDA and EMA stating clearly how they look at this. So to me, it looks a bit ad hoc, and I think it would be great to get more transparency about how much of evidence will be needed depending on the population size or other such factors. And the results we have are showing that regulators should be more liberal for everything else being fixed. They should be more liberal for very rare diseases and more conservative, comparable for common diseases. And kind of the same when we're looking at the payer, that it is rational for the payer to pay more for rare diseases than for common diseases. There are some arguments in the literature that it would be unfair to pay different amounts for drugs for rare and common diseases if the drugs have the same efficacy. So take two patients. A has a common disease, B has a rare disease. They are equally ill. They have drugs that could cure them or to give the same amount of health benefits to both of them. So why then are some people saying, why should the society spend more money on the patient with a rare disease and a common disease? Shouldn't that be completely a factor that should be factored in? And I think that's a reasonable argument in a static model. But what we're showing in a more dynamic model is that, or what we could model is that these kind of incentives will have different effects depending on if the disease is rare or common. Because for a common disease, you have a huge market anyway, so sponsors will likely develop drugs, even if they have borderline significance in phase 2. They may be good, but perhaps they are not. But for very rare diseases, unless we have clear incentive structures, then sponsors will tend to terminate all drugs unless they have excellent efficacy. So moving on to bullet point 2 here. We are saying that the society should factor in how rational agents are acting to find the optimal roles. We also have seen that transparency in the societal roles will translate into a loss for society, for the patients, for the sponsors. And it's understandable that that the regulator cannot say exactly how to react on every potential data set. And there could be other factors, so unexpected safety issues. So if you start with, do you think that one efficacy variable, one safety variable are important, then when looking at the data, unexpected things could happen. Regulators may be aware of data from other compounds, and they may factor that in. So it's understandable that total transparency isn't possible, but the less uncertainty we get in regulatory and pay rules, the higher the value will be to everyone. The optimal designs, the best designs for when we have different subpopulations, it depends on a number of factors, like the prevalence of being biomarker positive, and the efficacy prior. How much do we believe that the drug is working better or much better in the biomarker positive subpopulation? But it's possible to model these kind of things and find an optimal. And that optimal could be different from the sponsor and the society. So trying to align these perspectives and factor in how sponsors will act could be very important. So everything else fixed, it is optimal to society to lower regulatory requirements and also have a larger willingness to pay for orphan drugs. And that is more or less ending the talk. I would like to give you a number of references, and you will be able to find this presentation on Ideal's web page, so you could check out these references. And on the Global Ideal web page, you can see also a lot of other publications. I think we have some 40 peer-reviewed articles already by now, and more are coming. The Ideal web page you can find here. So thank you very much for listening. And if you have some questions, I'm happy to take emails, and I will try to answer to the best of my ability. Thank you very much.